Good afternoon or morning if you're on the far coast. Um, my name is Joshua Fortreed, and I'll be leading this workshop on the LungMAP ingestion broker, and we'll be going over how to submit and curate the metadata for the different submissions and projects and data sets that you'll be submitting. On the call with me is my close coworker, Kevin Burns. So he'll help me with this and help answer a lot of questions. We also have different members of the DCC team here, just in case there are other questions that need answered. During the presentation, please stop me if you have questions. I will also try to monitor the chat. If my DCC colleagues can help me monitor that, that would be helpful. It's just hard to navigate sometimes with all of it. But if you have a pressing question, please unmute yourself, ask me. If it's not really a pressing question, you can put it in the chat or interrupt me if needed. With that said, we'll go ahead and get started and see if more people trickle in. So the first thing I want to go over is some of the goals of this workshop. And one of the main things is we want to explain the metadata entities and how they connect with the different metadata that you're going to be putting in the ingest broker. And this goes all the way from the donor to the final analysis files that you generate. There's multiple steps between the donor and these analysis files, and these steps include the donors, the tissues, cell suspensions, files, protocols, and each of these steps are then linked via one or more of these protocols, as you'll see. And so we kind of want to explain this in a high level view, and then get into the ingest broker and actually show you. We'll demonstrate the ingest broker tools, and that includes both a web uploader and a CLI file uploader for bulk loading of lots of files or big files. And then two different curation tools that we have. One is a web form, which is a single entity curation tool. And then we have a multi-entity spreadsheets curation tool that we'll show you how to use. And then we want to explain the benefits of each of these tools and when to use them. For instance, the CLI file uploader, again, is for these large files like FASTQ files or any files housed on a server, as opposed to the web uploader, which would be for smaller files that you have access to. Explain the difference between the web forms and the spreadsheets, where the web forms are really used more for details about the submission and for curating the protocols, whereas the spreadsheets are really used for everything else. So briefly, we want to again talk about the metadata for all of the stuff that we'll be putting in the ingest broker. So I mentioned that we all start with some donor. If you're getting tissues from Brindle, then your real start might be at a tissue level. But ultimately, we're starting at a donor. We're going through different processes to get to a tissue. If it's single cell data, then we're going into single cell suspensions. From the cell suspensions, we might run multiple sequence runs out of them. We might get different aliquots out and run them, and then from that we'll get FASTQ files, and then from those FASTQ files, we're going to perform some analysis. We might get some file like a H5AD, and then if we're having multiple donors, we might combine those into secondary analysis files. We're combining those all together. And so this is really what the graph looks like, is, going from the donor through a process that has a protocol attached into the tissue, and continuing on the graph all the way down to the final analysis files, which are really the meat of what you're getting at. When you take this and you look at a whole project view or a whole submission view, then it starts to look something like this, where you have each of these different donors out here on the edge of this wheel and spoke display. And then from each of these 7 donors, we're going into tissues, cell suspensions, different numbers of sequence runs that we're doing. So here we have 8 sequence runs that were done, giving 8 different sets of FASTQ files. But for this donor, we only did 3. And so you can do that, you can curate that. Each of these are going down to a single H5 file that represents all of the information from these different sequence runs. And then eventually all of these 7 different analysis files are combined into one set of final analysis files that are really comprehensive over the entire submission. It might be an H5AD file. It could be a CSV file with extra information. It could be UMAP coordinates for that H5AD file, and maybe a gene set. And so this is what we're getting to build. And so the steps for this curation are fairly simple. The first thing we do when we get into the ingestion broker is we create a new submission, and then we curate the details around that. That'll be like a submission name, who has access to the submission. Are there any publications coming out of the submission, and what are those publications? You can put in a quick description or abstract for the submission, contributors, and more. After we create that submission, really one of the next things to do is to start uploading your files. So these are going to be your sequence files, your analysis files, readmes. For submissions that include lots of imaging data, currently we don't have the workflow set up for exactly how we're going to ingest those, whether they're going to come directly through the ingestion broker, or if they're going to go directly into our imaging server, which is Omero. So for now, and for the purposes of this workshop, we're going to exclude those sorts of imaging files from the process. After we upload the files, we curate them, so just basic information about the files. And then we start to create and curate all of these other nodes that we have to do, the protocols, the donors, the tissues, for single cell data, which is a lot of the data, these cell suspensions. And once we have those, then we can annotate our sequencing runs, which basically connects those cell suspensions back to those sequence files that we uploaded. And then we can further curate to go from the sequence files to analysis files. And then just some basic notes for the curation. So while I did give that last list in order, the order is not really strict, but is more an organized workflow that can be beneficial. And one of the reasons it was outlined in that order is, so if you create and curate your donors before creating the tissues, it allows you to, once you go and you start curating your tissues, because you already have the donors in your submission, then you can very easily link the tissues to that donor. And so it's starting to build the connections in that graph. Another thing is curations can be saved at any time, and this allows for partial curations. So as you're going through and you're filling out the information, if there's some required information that you don't know, or you don't have all of your donors for your submission readily available, you can go ahead and curate the information that you do have. You can always come back later and curate any missing data. And then finally, some of the curations are done via spreadsheets, others are via the web forms. And I'll reiterate again, the web forms are primarily used for the submission details and for the protocols. And that's because these are more one-off items. You only have one submission detail per submission. And for protocols, you might only have one collection protocol or one sequencing protocol. Whereas for donors or for tissues, you could have dozens or even hundreds. And so curating those in spreadsheets allows you to very, very quickly curate those in a uniform manner. Before I go to the live demo, I just want to give you a quick overview. So within our ingestion broker, this is what a submission page might look like. And I've edited out and condensed a few things, but just to give you a overall topology of it. The right-hand side is very simple. You have two areas right now. One is just a file summary, which gives a basic count and file size for the different types of files that you have, as well as links to go to the web upload or the CLI upload, which I'll show you in a minute. And then you have this new forms dropdown, which allows you to create new protocols or donors or things like that. And then the left side is the main meat of this page. So you have submission details up here. You'll have information on which users within the ingestion broker have access to the project, who are the contributors, the funders, the publications. A lot of this is detailed in the submission details when you edit it. There will be a box for file details. So we'll list the last 10 files that have been uploaded. It'll give the path, the file name, the size, who added them and when. And then there's links in here to either view all or to start some of the spreadsheets for curating them. And then down below the file details, you'll start getting into the information about any donors that you've added to the submission, as well as a link to the spreadsheet to add more. Information about the protocols, and you can click to edit them. And then information about any samples, which we're defining as any tissues or cells. Then there'll be links to the cell spreadsheet and the tissue spreadsheet. There'll also be a little more information at the very bottom, but we're not going to go over that at least right now. So from this submission page, there's links to the web upload. The web upload will just take you to our S3 bucket uploader, where you can, via this, just click upload, find the files in your computer and upload them. Very easy to use. If you're doing it from a server, or if you want to do it via the CLI uploader, when you click the CLI upload on the submission page, it will bring you to a page that looks like this, and you'll, depending on if you're on a Unix machine or a Windows machine, you'll copy the text given here into your command line, and then you can run the commands to start the upload process. Now, I've conveniently blurred out any access or token keys here just for security purposes. If you have any problems with any of these uploads, please let us know and we can work through them with you. And so at this point, I'm going to move to the live demo of the ingestion broker. But before I do that, I just wanted to open it up for any questions that anyone might have. No questions? Okay. And again, for anyone who has joined since the start of this, if you do have questions, please stop me, let me know, and we can answer them. So to get to the ingest broker, there's ingest.lungmap.net, but an easier way might be if you go to the LungMAP website and go under resources, then you can, is this correct? No, that is not the way. A DCC member who is on, can you direct?
That was correct what you were just doing. If you go back there.
Ingest data?
Yep, and then you just
here's the information here.
And yeah, and you can click on those links, or you can but you would check on the long-term ingest broker. Yep.
Excellent. So here's the information. Yeah, so if,
and you can also just go up to the top where it says submit here. We're at the very top of the page.
Top of
LungMAP Consortium Ingest Broker, submit here, the very first line.
Oh, very good.
Yep.
And then this document is good because it will give you information on how to request an account. So we do this all through a single sign-on. And so you can email these email addresses and get your account set up. So once you get to the ingest broker, there will be lots of submissions that you'll see. I'm going to go ahead and move to our dev site, just because as I'm going through this and I'm adding data, I don't want to pollute our production or cause any issues. And so when you log in, you'll see any submissions that you have access to. You'll get just the short name of the submission, who's the owner of it, when it was created, if there's any issues with it or not, and then a details link, which allows you to go in and edit the submission. If you don't have any submissions or you need a new submission, then you can click new submission. It will eventually, in our production environment, it'll come up with this ingest disclaimer, just talking about the different rules we have and not to upload any sensitive or PHI information. Click acknowledge. And so here it's generated a new submission, and then you can go in and edit it. For this purpose, I've already created this submission here that we will start looking at. So once you're in here, this should look very familiar, because this is basically what I just showed you in the slides. So you'll see a little bit more of the submission details. I edited out, and then you see your authorized users, some more core information about it, contributors, funders, publications, and then you get down to your files and your donors. For this purpose, I'm not going to actually demonstrate the web upload and CLI upload. If you need help with that, please let us know, and we can work on that with you. And so because I already have the files uploaded, the next step I want to do is I want to go through and curate them. Most of these I've already curated, so I'll just do a few. So because this only shows the first 10 files, I'll click view all, and I can see all of the files that are in here. One of the very first things you'll have to do is, if a file does not have a schema type associated with it, you'll have to go over and click this edit schema link. And this is fairly easy to do. Basically, we're trying to identify what type of a file is it. Is this a sequence file, analysis file, image file, a reference file? And so I know that this is a FASTQ file, so I'll click sequence file and click save. It will ask me to confirm. I'll go ahead and click confirm. And now it shows up there. And then from this view, there's lots of different spreadsheets that can spawn from this. I can edit all of my sequence files, I can edit my analysis files, or I can edit the sequence runs. And so what I'll do is I'll just go into the edit sequence files, and this is the spreadsheet that you will see. So the spreadsheet contains all of the columns of different metadata that we look at for these sequence files. If we went to analysis files, you might see slightly different columns. These columns can either be free text, or if they might have a little drop down in here, and so you can double click and see the different options, and you can change it if need be. I'll change that back because that's incorrect. If you're not sure what a read index is, you can just mouse over and it's going to give you some information on that. So anytime you see this little question mark here, there is a popover README that will give some information and then some examples. So for instance, in this column, an example of 51 for a read length. And so this is pretty self-explanatory. Going through any required fields will have this sort of a red asterisk next to them, just prompting you that eventually this field will need to be filled out. But if it doesn't have that, it's not technically required, although the more information, at least the more accurate information, the better. So when I started this, there was that one FASTQ file that didn't have a schema type. I've set it to sequence file, and so now I want to scroll down and try to find that, and it should be fairly easy to find because there should not be some information in here, although I am not seeing that one. Might be a slight bug. So the other thing I can do is I can say double click on a column, and from there I can sort based on that column. And so one thing I can quickly note here is that this index file has a read length. That should not be correct, so I can delete that out. And I can scroll down and see if there's any other issues. So there's my index ones, here's my read ones, here's my read twos. If there's a very specific file that I am looking for, I can go into the top right and I can search for that. So say I'm looking for a file for donor 519. I can search, I don't see that. Let me try 511, and then I can see all, several of these that have 511 in there. Or if I'm another great use, depending on how you name the files, if you're looking for all of your read ones, then you can filter based on _R1. If that's how you've named them, it's going to bring up all of my read ones, and then I can very quickly curate them. So I could select this as read one, and I could take this and I could drag it all the way down and make sure it's populated. Are there any questions on using these spreadsheets? Or at least this specific spreadsheet. One thing you can do, so after you've curated some, to save, you just want to go up here and click save. You'll know it's saved because it'll actually refresh the spreadsheet. If it doesn't refresh the spreadsheet, that means that something went wrong, and please contact us and we can work through it. You can also download the spreadsheet as a CSV file. And then when you open it up, you can actually go in and edit some of it and then re-upload it. For instance, if I needed to change all of these read lengths, or maybe I just want to change these top ones, I could do that. I could save this and then re-upload it, and it will put that information in there. I won't do that right now just because I don't want to put the wrong information in. but you would click the upload button here, go through, find the file, and then click open to upload it. The upload does not work for adding any records or files to the spreadsheets. That's just because, for files, we need to make sure that we actually have the files in our system so that we can curate them. And so that doesn't allow you to add any files to this. So now I'm done with the spreadsheet. Everything looks okay to me. I'll save again just to make sure, and then I can return back to the product page. If I needed to edit the analysis files, I can go down here and click Edit Analysis Files. And now you can see that there is different columns here. So file format, but then we have matrix cells. So if this is a matrix file, I know how many cells are in the matrix. Again, you still have your file source, content descriptions, different columns. One thing that this spreadsheet has that the last one did not have, though, is an analysis protocol column. And so this column is actually going to read from any of the analysis protocols that you've already set up within the submission. If you haven't created any analysis protocols, then there won't be any values for here. And so if you're curating this, you'll just have to skip this column and come back to it. The other thing that this has is parent files. So any analysis file is going to be generated from some parent file. So if I clear this out, I can go ahead and curate this. So the sequence file here is for individual 507. And so what I can do is I can double click in here and I get the list of all sequence files that are in the project. But I really don't want to scroll through here and try to find all of the 507s. So what I can do is I can start typing in 507 and it will filter this list to any that have 507 in it. And here you can see that there are sequence files for 507 and there is an analysis file for 507. I want to select the sequence file, so I can just go through, click all of them. You'll notice that this is a multi-select. Once I've clicked all of those, I can click done and it will populate that. And so this is how you generate the link between the sequence files and this H5 matrix analysis file. You'll see this feature on most of the other spreadsheets. Do that. We can scroll through, make sure everything else looks okay. Perhaps I'll double click on file format just so I can group all of the H5s together. And I can scroll down and that's an H5. I notice it doesn't have any parents for this, so I can go through and put that information in. So I'll just double click here. This is 502, so I'll enter in 502 just to limit, go through, click those. It's again the FASTQ alignment, so I can either double click here and click FASTQ alignment, or I can drag this down and fill it out. We have the H5 in here. These are looking okay, H5, H5. Here's an issue. This is actually a text file. We've called it an H5, so I can quickly change that. And everything else seems pretty good. And so I can go back up. I can save this, make sure all of the changes I've made are in the system. Now I can go back to the product page to start curating more. So now that we have the sequence files in, we've done some of the curations on them, we can start on donors, or we could start creating some of the protocols. So we already have some protocols in the system. And in fact, I think these are all of the protocols I need. But I can go ahead and just create another dummy one just to see. So if we have to create a new protocol, we come up to the top. We're going to select what we need to do. So we want a protocol. And let's go ahead and click Analysis Protocol, Create Form. This is going to bring us to the web forms. Eventually this will get cleaned up a little bit. But for now, we just want to give it a protocol name. So test analysis protocol, this description. If you have your protocols published, either in a publication or on protocols.io, you can put the information in here for that. And then different protocols are going to have a few different fields. These ones should be very similar, but then they might have additional tabs. This is an analysis protocol, so it has an analysis tab. And so from here, there's a few different fields. So we have some computational method. So we'll just put in 10x for this. When you get fields that look like this, where you have three fields that look very similar, in the middle one will say ontology label, and then this will say ontology ID, and this is then what we're going to clean up. What you want to do is you want to come to this one first and start typing in, because this is coming from a controlled vocabulary, and as you start typing into here, it's going to start giving you a drop down of different items that are appropriate for it. And so I know I picked analysis protocol, but if this were, say, some dissociation or something, I can start typing in here and it will start coming up with information. If you are unsure what a term is, you can mouse over it. And if there is a definition associated with it, it'll give you that definition. One of the great reasons to start with this field is because, one, it is using the controlled vocabulary, but also if I click this, then it's going to go ahead and fill out all of the other fields that are needed. So I've done that. If there are derivation processes, I can go through and I can click that, data normalizations. I can fill out that information. When I'm happy with all of the information, I can go ahead and click save. And now it should have added that in my protocols down here. Are there any questions on filling out any of the web form there? If time permits, I can go and show you a different protocol, or I could even show you one of these other forms. But want to go through a few more of the spreadsheets. So now that we have all of our protocols, and again, one of the things that the protocols do is it helps us connect the different nodes. It helps us connect donors to tissues, tissues to cells, cells to sequence files. And so curating the protocols, at least giving, creating them as small little things, and then going back and filling out all of the information for it later is very helpful. So I have these protocols generated. So now I'm going to go and I'm going to create a new donor, because I did more work and a new donor came in, and now I need to put in that donor. So I can go and I can click Edit Donors. This is going to bring up my donor spreadsheet. And the donor spreadsheet, I believe, is the biggest spreadsheet by number of columns. And so here you can see I have a lot of donors already created. So here is the donor ID. There's a description of them, a controlled vocabulary for species. Again, this is a type ahead. And then lots of other columns. We've tried to group the columns by what sort of information they provide. And so here you can see these yellowish columns are a little bit more about demographics. So the sex, the age, developmental stage, ethnicity, if these are human samples, the strain, if these are a mouse sample, gestational age, height, weight, BMI. So some of these are crossed out based on other columns. So because these are not human, then you don't give an ethnicity. And because we don't have an age, you can't specify an age unit. So if I were to go in here and put an age in for these, say this is 23, then this cell becomes active and I can decide if this was 23 days or 23 hours. So I can put that information in. You can continue scrolling to the right, or you can start to toggle these groupings off. So if I'm done with the demographics, I can toggle it off. And then I get this section here, which is information about death. So cause of death, cold perfusion, organ donation types. Again, a lot of these are controlled vocabularies. I can start to toggle on some of these others. So say medical history, especially if it's human, and then any accessions. If you have accessions for these in biosample or HDBR or any of these others, you can put in the accessions for these donors. I don't have any of that information. What I want to do is I want to go and add a new donor. So to add a new donor, what we're going to do is go to this donor lookup and add, and I can start typing in the donor ID. Eventually this will start to do a lookup. So if you're using the same donor between multiple projects, we'll have it so you can import that donor. Also, if you are using Brindle donors, we'll have it so you can import that information and you don't have to put it all in yourself. We'll go and click add. This as a new entity. It'll add it down at the bottom, and then I can start filling out the information. Because a lot of this is the same, I can just drag down, or again, I can do a type ahead. And one note about the type aheads is where possible, it also uses synonyms. So if this was a human, I could start typing in human, and it will bring up Homo sapiens, which is the correct terminology. Or the fact that this is a rhesus monkey, I can go in and type in rhesus and get the genus species name for it. So I can do that. I can start to copy down. I believe this one was a male, and we can just continue to go through. Again, at any point in time, you can click save. So it's saved, it refreshed. This donor is now at the top just because it sorts in alphanumeric order. And then I can put in any more information that I need to. But for now, I'm happy with this. I haven't made any changes since I saved last, but I'll just save again just to make sure. I can return to the product page, and then I can go and start creating some of the other things. So now that I have the donor, I probably want to have some tissues from that donor. And so I can go to the tissues sheet. And so here you can see kind of how this is set up. So you have a tissue name similar to how we had a donor. You specify where this tissue came from. Did it come from another tissue, or did it come from another donor? And this will give a list of all of the donors and tissues that are in your project. And so you can see those. You'll select a sampling protocol because this is a sampling, and so you can select the sampling protocol and then start to fill out some of the other information to the right. So what I need to do is I need to create a dissected lung for this individual. So I'll go ahead and type that into the tissue lookup and add, click it, come down here. I can either click that, or I can start typing in 502 to limit if I have a lot of selections. I'll go ahead and copy this down. This should be the same, so I can copy these down. I can copy multiple down if I need to by highlighting more than one column and then pulling it down. And then I can click save. Now this should be in here. Oh, for some reason it didn't save the parent tissues. That's a good check. So I'll go back in and do this again. And let me click save just to verify. And there, it took it that time. So I'm happy with this. And then I could go on and fill out more information, or I could return to the project page and go on with the next sheet. So after I've done tissues, because this is single cell, I'd most likely want to edit and create some cell suspensions. This is going to look very similar to the tissue spreadsheet, where you have your main entity here, your cells. You have where those cells came from. Was it a parent tissue, or did you take one set of cells and split it in two? Again, what was your sampling protocol that you used to do this? And so here you have a selection of any collection protocols or dissociation protocols, and then lots of different fields relative to cells, such as estimated cell count in your sample in the species. If in this you have subsetted for a particular type of cells, say epithelial cells, you can start that's not how you spell epithelial. You can start typing in and get that. If it's macrophages or something like that, you can start the type ahead and then select specific type of cell that you have selected for. If you cannot find the specific type in this column, a lot of times there's a column that has a specific at the very end of the name, and you can fill out in there. So if I couldn't find a particular type, then I could put in that these are alveolar macrophages. And I could save that information. There aren't any other required columns in here, or required fields. I now have that information, so I can just go with this. I can click save if I want, and then return back to the project. And so what you can see is very, very quickly we've been able to add a donor, add a tissue, and link the tissue to that donor via a collection protocol. I didn't go ahead and add cells, but it would have been very easy to add cells for that donor, and then or add cells and link them to the tissue via a dissociation protocol. And now that I have my cells, what I want to go back up to the files and click edit sequence files, or nope, not click edit sequence files, click edit sequence runs. And from here, what I can say is, well, this is my sample. So these are my cells. This was my sequencing protocol and my library prep protocol. And then these are the files that I generated from it. And so if I want to add a new record for this, instead of a search box up here, there's just this plus sign for adding a new record. I'll scroll down to the new record, and now I can select a particular cell suspension. So say I'm doing 505, I can click that. I can copy these down, just like I have on the other spreadsheets. And then I can start picking the read one and read two files. What makes this a little easier is because we've already curated our files and curated what's a read one and a read two, we've now limited this this column to only read one files. And so when I type in the 505, I only get my read ones, although here you see that I have two different ones, one for lane 1 and one for lane 2. So I'll pick the lane 1. Same for read 2. I can type in 505, pick lane 1. That's based on the naming I have there. So lane 1, read 1, lane 2, read 2. And then because I know these data, I know we have index files, and so I can toggle on my index files. And I know I only have a one index file, and so I can double click in here, type in my 505, and get the index file for that. And so now I have this curated, and I can click save. I'll add that. And so now we've been able to go from the donor all the way to the sequence files. And previously, when we were looking at the analysis files, we were able to go from the sequence files all the way to the analysis file. And so in a matter of maybe 20 minutes or so, we're able to very quickly go from the donor all the way to the analysis.
Hey, Josh, before you leave that,
Yes.
Just note real quick, because you had the multiple lanes there, that there's actually 2 records for each of the samples. So you'll notice 512 repeating and 509 repeating. That may be a little tricky once you're in there wondering, where do I put, because you have a multi-select function in a lot of these cells. In this instance, you want the lane separated and you just want to duplicate the sample name.
Correct. And that's a very good point. So especially if you're doing multiple lanes, you're going to have that. So again, you see this one sample, 513, was run on 2 different lanes. And this record down here is for lane 1. You see L001, L001, L001. And then you'll see the lane 2 up here. One good thing to note on this is this does not allow you, so this particular dropdown does not allow you to do multi-select. So where multi-select is valid, if you have one selected and you click another one, it will select both of them. But in this case, because only one value is allowed for this, it does not allow you to select multiple files. But it is very important to notice, yes, that for a lot of these, because there are multiple lanes done, you'll see the records multiple. It'll look like the records are done multiple times, so it's just for different lanes. Okay, so that's a majority of it. I guess one thing I did not show is editing the sequence details. So at the very, or the submission details, at the very top, there is an edit button here that you can go to edit the actual submission data. So you can put in a project title, a very short label, a description for it. Then there's these other tabs, so you can start to add in contributors. You can add in multiple contributors. You can select different roles. Are they a principal investigator? Are they some computational scientist, or they a regular investigator, a data analyst, data curator, or coder? Publications. Again, if there's multiple publications, you can add them in here, putting in their PubMed ID or a publication DOI, or just a regular URL, as well as a title. Any funding sources, very good to put in. So that will show in the different systems, and you have a record of them. If there's other accessions, say you've deposited in Geo or BioStudies, Array Express, you can put the information in there. And then if there is some supplemental data, reach out to us and we can help you with adding that in here. But there isn't there, and so I'll just go back. And I'll just go back to the previous page. So that's pretty much the ingestion broker. Sorry, I've used up almost all of the time. One thing we are working on is is very good documentation on this. So as you're going through this, if you're having troubles, you can reference the documentation. The other thing is if you're going through this and you're having any trouble with it, errors are coming up, you're not sure what information to put where, please reach out to us. We're more than happy to get on a call with you to help and to help curate some of this. You know, we're really here to work with you to try to get the data submitted in a uniform way, in a consistent way, in a verbose way, so that as we move this data into different data brokers and into the LungMAP website, we have all of that rich metadata that allows for good searching. So at this point, I think I'll open it up for any questions, and I am more than willing to stay on as long as we have this, you know, we can keep this Zoom call going. So if there are questions, I'm more than happy to stay on past 3 to answer any questions. People do have their speakers turned on, right? I'm not just talking to no one, right?
No, it was great, Josh. Thank you.
I'll second what Surya said. Fantastic work.
Yeah, I'll jump in as well. As a person who has submitted and worked through this, I do like some of the newer tools. I think that'll be helpful, but I think this is a good overview. I'll be submitting more, so I'm going to probably have a question. It's always hard when you're looking at it until you really do it. But like I said, the previous early experience wasn't too bad. I'm hoping that this will be a little bit easier.
Yes, the addition of these spreadsheets should really help with a lot of the submission work, because it really does streamline it. Because previously you had, you know, if you had to add a new donor or a new tissue, you had to go up here and add a new tissue, and it would, so for a tissue, it would take you to this view, so this is something I didn't necessarily show. So let me go back just to show you that. So within all of these different forms, you can click edit to go to a spreadsheet view of this, but if you want to go to just a web form view of it, you can. So clicking the edit button here will take me to a web form view of this donor. So here's you know, donor 50219, and so I can go through and I can see the fields related to death, the fields related to disease, the demographics, medical history. So a lot of these are very similar to what you saw in the spreadsheets. And this does have, you know, a lot of the same functionality, but here you can only curate one at a time, and it might be a little bit harder, whereas the spreadsheets, it allows you to enter in, you know, a dozen at a time and then easily curate them. So yeah, the spreadsheet should help.
Josh, I was just gonna mention a couple things that I think people have brought up before, is that you do not, for a entry within the ingest broker, it does not have to be a nice, neat packaged paper. It can be a broad collection of samples that you've collected that you don't know where they're going and when. And we encourage submission early and often, even if you don't have a paper in mind. We won't push anything to the data browser until you authorize it and you finalize your submission and talk with us. Also, if you're planning submissions, you know, feel free to reach out to us. We can walk through the actual submission of your data set with you from a data wrangler perspective. And finally, if you'd like to see what these data look like once they've, you know, so Josh showed you all this nice metadata here in the files. If you'd like to see what these final files and metadata look like, you can go back to the lungmap.net website and to resources and data browser. and you can see what the translation of this information looks like. So for example, if you go to that last one, you'll be able to see, how this project description information that you added there, how the metadata that you added at a high level is there, including, which contact, who's the contact PI, what publications are associated, the contributors, the organization, and how these files are downloadable. And so this is kind of a pretty view of what effectively all that metadata that you put in looks like in the end.
Thank you very much for that, Nathan. And I'll reiterate the fact of, don't wait until you're ready to publish to get the information in here. The sooner you have information, that's when I would put it in, or if you're waiting, for your samples to be sequenced, or you're waiting for analyses to run, or a meeting gets canceled and 15 minutes shows back up on your calendar, just log in and add a little bit of information. I imagine all of us have gotten to the point where we've gotten near the end of a project and we've needed to submit data, and we're struggling to go back through notebooks and files, and is the file on this computer or on that computer or on a server or on my laptop, or which flash drive was it on, and it's hard to find the information. And the best time to enter the data is when you have the information nice and fresh and right in front of you.
And to reiterate another point here is this is kind of the jumping board for having, if you have human study information, human sequence information that needs to go into dbGaP via LungMAP, having the data and the metadata all in the ingest broker is the first and most important step. And then everything else is streamlined from there once everything is organized in terms of the dbGaP submissions. So please reach out to us separately if you intend this to be a dbGaP submission, and let us know. And again, we'll work with you. And finally, as noted in the chat, this a video recording of this of what you've seen here will be put online very shortly and sent out to everybody here and made publicly available.
And we are continuing to work on the ingestion broker. We're adding new entity types. So I know there are some projects out there with organoids. And so we'll need to add that as an entity type and create spreadsheets for it. We're also looking at ways to streamline this a little bit more and give you some more hints about where you're at on the status of the project, and maybe even easy link outs to ask for help. And so we're continuing to improve this, to add more functionality, refine functionality that is there. And so as you're going through it, if you're noticing that something's taking a really long time, please let us know, and we can see if we can change how it's done to make it easier and faster for you, as well as more robust. With that, thank you for your time. Like we've said, we've recorded this, and we will be sharing the recording with you, and then you can share it with whomever you need. Please reach out to us if you have any questions as you're working through stuff, and we're more than happy to work with you and jump on calls if need be to work through any problems. Thank you for your time.
Thanks, Josh.